Hello everyone. Today we'll see the topic PCR, that is polymerase chain reaction. What is PCR? It is actually a chain reaction which is very quick, simple, and it is most accurate molecular test to create the unlimited copies of DNA from just one single strand of DNA. Okay, so this PCR technology we can use for genetic testing or for forensic purposes also. Now what this consists of? It consists of the PCR cycle. Okay, now what exactly there is requirement in case of the PCR? So these are the PCR components. So first of all we have to prepare the master mix. Okay, so when we are going to prepare the master mix, we have to take such types of appendrop tube. Into this appendrop tube we have to add the DNA sample. Okay, that is our template DNA. Then we have to add the primers, nucleotides, then buffer and the tag polymerase. Okay, now how to add this? See, suppose we have to make the master mix of 30 ml, then and we are going to add 1 ml DNA sample. Okay, 1 ml primer, 1 ml nucleotides, 1 ml tag polymerase. Okay, and 10 ml for example buffer. So 10, 11, 12, 13 and 14. So 14 ml we are going to add the other components. So how much remains? So that is actually the 16 ml. We need what? We have to add the water, or these components can be adjusted based upon our requirement. Okay, now after addition of all these components into the PCR tube, we are going to keep it into the thermal cycler. There are various slots available into it and you can run the PCR. What this PCR cycle consists of? It consists of denaturation process which takes place 90 to 95 degrees Celsius at this temperature. Then annealing process is there where actually the primers they bind to the template, and further extension. Now this is very important temperature, that is annealing temperature. How it is decided? That depends upon your primer. What is the requirement of annealing temperature by your primer? You can test that by running the gradient PCR. Gradient PCR. Okay, so in gradient PCR, if you can see that this, it has having the different slots. Okay, so in different slots you can adjust the temperature. For example, here 55 degrees Celsius temperature is there. You can take it as a 50 to 55, means you can take 50, then 51, 51, 52, 52.5, 53, 53.5, 54 and so on. And then at which temperature you you get the amplification, that will be your annealing temperature. Suppose the given annealing temperature for your primer is for example 51, but you got the amplification as at 50 point somewhat 5 degrees Celsius, then that will be your annealing temperature at your lab. Okay, so in this way you can decide annealing temperature of your primer. So after the PCR amplification, we can check that for the quality and quantity purpose. Okay, these are the advantages of PCR technology. That is the it is highly specific. This PCR distinguishes the DNA sequences just by one nucleotide, and that make it very accurate techniques. Then it is very sensitive because a small amount of DNA though you have, it can amplify or use the multiple copies of it. It is versatile too because it not only used in the genetic testings, it is also used in the criminal investigation, paternity testing. Okay, it is very rapid and efficient, that you can get multiple copies of millions of copies of DNA from the small amount of DNA sample. Then these are the disadvantages of the PCR technology, that is contamination. There shouldn't be any contamination, otherwise there may be the chance of amplification of different DNA. Okay, if there is contamination, then cost, it is cost effective because PCR machine is expensive. The materials, the tag polymerase, the master mix all the components required, they are expensive. Lack of novel information means it amplify only the target's specific DNA and not gives us the information about the other sequences present on the DNA. Then inhibition from the sample component. It may be possible that sometimes the heme from the blood, it can act as an inhibitor, or the alcohol, if alcohol remnant if there is in the extracted DNA. Okay, so that can also be the acts as an inhibitor for the PCR. Errors in the amplification means if there is the addition or deletion of any base substitute is there, again there may be the false possibility of false result. Overall PCR is very good for the research purposes, but there is need of an expertise. Okay, these are the references, and thank you.